In most living creatures, cognitive abilities, physiological processes, and behaviors vary through the day and night, which introduces a potential time of day bias to data collection. Most lab-based research participation usually happens during regular working hours, from 9 to 5-ish, while online survey-based research is more flexible and can happen at any time during waking hours. If the behavior of interest varies, this needs to be considered in both types of research. Probably the most obvious time of day effect is the sleep-wake cycle. Unless you're studying sleep, it's best to collect data when an individual is awake and alert. For diurnal humans, that's the day, but most rodents are nocturnal, meaning they're active at night. This introduces an important consideration when using non-human animals like rats to model human processes, but many studies test rats during the light phase, when they're least alert. Of course, it's difficult to work with rats in the dark, so one solution is to house animals on a reverse light-dark cycle, so their active time matches the researchers working with them. Dim red light provides low illumination to mimic night, but allows researchers to see. Tests of humans are usually done during the day, but there are variations in behavior, alertness, and activity from early morning to evening. This study compared age differences in optimal time of day in university students aged 18 to 22 with older individuals aged 66 to 78, recruited from a research center on aging. They completed Horn and Osberg's morningness-eveningness questionnaire, a survey designed to determine the degree to which people are morning birds or night owls. Data from the questionnaire was divided into 5 categories. The younger group were more likely to prefer the evening, whereas the older group were biased toward the earlier part of the day. In Study 2, researchers recruited participants from Study 1 to test whether individuals actually performed better at their preferred time of day. The experiment involved a verbal memory test, conducted either in the morning or afternoon. What they found was congruent with Study 1. Younger people performed better in the afternoon, as shown on the top row, while the older people performed better in the morning. Testing these participants in the afternoon would result in huge differences and different conclusions about cognition across age groups. Time of day effects are also easily demonstrated in laboratory animals. 2 examples are shown here for commonly used C57BL6 mice. The graph on the left shows average activity across a 60-hour period, while the graph on the right shows average heart rate. The light and dark bars at the top show when lights were on and off, and it's clear that activity levels and heart rate are highest during the dark phase. Not surprising, since these are nocturnal animals. Cyclic changes in behavior and physiology throughout the day vary by mouse strain, so this can also affect research results. This study looked at the tail flick test for pain, shown on the left, where the animal's tail is placed in the path of a heat source, and the time it takes to remove their tail from the heat is used as a measure of pain sensitivity. This is displayed as latency to flick in seconds, and it's shown in the graph on the right. The longer the latency, the lower the sensitivity to pain. This study compared pain sensitivity in the light and dark phases for 3 different strains. C57BL6 mice, shown in dark blue, I29S1s, shown in green, and F1 mice, a hybrid of these 2 strains, shown in red. During the light phase, the 129S's showed the longest latencies, indicating lower pain sensitivity compared to the other 2 strains. However, this strain difference disappeared when the animals were tested in their dark phase. The tail flick test is commonly used to test analgesic drugs, so the choice of strain used and the time of day of testing is important to consider. Test yourself. What is the best time of day to test participants? Explain your answer. The best way scientifically to deal with participant differences across the day would be to collect data over a 24-hour period. Of course, it's not usually possible to test human participants around the clock, and it requires specialized equipment and procedures to do this in laboratory animals. The most feasible approach for researchers is to be aware of whether there is a known time of day effect for their variable of interest, so that an informed decision can be used in choosing the testing time.